Hello everyone. So today we're going to be talking about multidimensional data analysis derived from tractometry. So as of now, you probably learned in the previous courses how to derive track profiles, for example. So here is just a brief recap of how do we extract these profiles. So first of all, we start with a whole brain tractogram for each of our subjects, and then we extract a set of tracks from that whole brain tractogram. Using various approaches. These could be automated techniques, for example, or via manual delineation. And then for each of these tracks, we define or we extract a center line or core streamline, which is going to be used later on to project different metrics. And this center line will, of course, vary for each tract. And then what we do is for each vertex along the tract, so each streamline is made of different vertex, we look at each vertex which point of the core is the closest to it, and then we assign it to this core. So we create these kind of assignment maps or label maps. And then what we do is simply map a certain metric on the track and then take the average within each of these sections. This can be a weighted average, for example. So depending on how far you are from the core, you might give less value to the metric. And if you're close to the core, you give a bit more value. And then you end up with these track profiles, basically, for each of your tracks and each of your subjects. But the tractometry philosophy is actually not necessarily based on track profiles. It's more of a general framework, which was introduced in ISMRAM 2011 by Sonia Bells, where, as they say, it's try to combine multiparametric data along multiple tracks. So in their abstract, and actually in the paper from Silvia DeSantis, they used tracked averages along multiple tracks, but they mapped various MR-derived metrics. So in this talk, we're going to be looking at tracked averages of multiple tracks in the first part, and then looking at track profiles a bit later on. And we're going to be talking about two concepts. So first, the dimensionality reduction concept, where we have a high dimensional space, such as having multiple tracks and multiple metrics per tract, and try to reduce the space into a low dimensional space, which is a bit more interpretable, hopefully. And the second concept we're going to be looking at is the idea of anomaly detection, where we try to look for outliers in our data. So dimensionality reduction. So typically when we work with multiple diffusion MRI measures in a univariate manner, we often have this statistical problem where each of these metrics are treated independently and tested against our hypothesis. And then we have this multiple comparison problem. The other problem by treating each of these metrics independently is that we totally ignore the relationship between these metrics. So they might actually be probing complementary information. So it would be nice to have a framework that could combine all of these metrics into some meaningful components. And that's what we're going to be doing today. We're going to be replicating a figure from one of our recent papers where we leveraged dimensionality reduction to create this new subspace where we projected different tracks and different metrics. And this was done in a cohort of typically developing children. And the nice thing is that these components, these new components, actually correlated better with age, so with the developmental age of these subjects, rather than any of the individual univariate metrics that form these components. Okay, welcome back. So we are now in the Jupyter Lab environment, and we're going to be replicating an analysis from this first paper here about dimensionality reduction of diffusion MRI metrics. So the goal here is as we will see, we will have a set of bundles with a lot of diffusion metrics that are mapped on these bundles. And we want to reduce the space of metrics. So basically, as you see here, we have 10 metrics, and we want to reduce this down so that we can interpret the results a bit better. So first thing is we're going to be loading our libraries. These are mostly Python libraries for plotting and for machine learning. And let's load our dataset. So as you see, the dataset has 792 inputs and 10 columns. So let's look at these columns. We have an ID column, a bundle column. So this states which bundle we're looking at. And then these are some features for this bundle. So the mean FA, the mean AD, RD, mean diffusivity, etc. So these are Dti metrics, and these are more advanced metrics derived from Hardy. So AFD is the apparent fiber density, NUFO is the number of fiber orientations in the bundle, AFD dot is a generalization of AFD, and FR is a restricted signal fraction derived from charmed. So as you can see, all these metrics may have some things in common, and some might pick up different part of the signal rather than others, but these are all features. And if you look at the data, here's what I was meaning. So basically we have our subject's ID, the bundle associated to each subject, and then the mean of each of these metrics. And this is repeating. So as we go down, the same subjects will reappear with different bundles. So for the ease of simplicity, we're going to select only a subset of these bundles for now so that we can better understand what's happening in the background and also for visualization. So let's pick like some main bundles like the corticospinal tract, the uncinate fasciculus, the arcuate fasciculus, the splenium and the genu of the corpus callosum, and the optic radiation. And I'm taking only the ones from the right side for now. So we have now this data set, and we drop the ID and the bundle features because we're going to be using only the macrostructural metrics. So we end up with 216 rows and 8 columns. First, we can look at the correlation between the features. So basically to see if there's any link between any of these metrics. And as you can see, we see some correlations between different metrics as we expect. For example, FA is highly correlated with AFD because these two are probing the anisotropy, let's say, in the tissue. And similarly, FA is also correlated with FR. This is also an anisotropy metric. So you can kind of see already, have an overview of different relationships between all of these metrics. So first, because all of these have different scales, so MD has a really low scale or values compared to FA. So we want to scale everything to the same order of magnitude. So this is done using the standard scaler and a scikit-learn. And we're ready to go. So first, let's look at the principal component analysis. So we're going to specify 2 components. So we want to reduce these 8 metrics to 2 major components. And that's it. That's pretty simple. But now we're going to have a look at what's happening in the background. So by looking at the explain variance attribute, we can see that the first component of the PC explains 48% of the variance in our data, and the second component explains 35% of the variance. So that's already a good indication that selecting 2 components accounts for almost 85% of the variance. And we can also look at which of our features contributed to each of these components. So you can see here I have 8 features and here I have 8 rows. So this is for component 1 and this is for component 2. So we can see that for component 1, we have metrics that contribute the most to this component related to FA, so the first one here, AD, RD, AFD, and FR, also AFD total. So these values are basically all the anisotropy measures. So we could call this component the tissue restriction or hindrance component. And the second one has mostly inputs from AD, mean diffusivity, and NUFO, so number of fiber orientations. So this PC2 is kind of different from PC1 in the sense that it could be named as the tissue complexity component, if you try to interpret a bit what they mean, let's say. So now we're going to apply this transformation to our whole data and look at this new subspace that we created. So the PC1 versus PC2 for each of the subset bundles that we've selected at the beginning. So first off, each point in each cluster represents one subject. and each cluster represents a tract, as you can see here by the color map. So if we try to interpret how these tracks fall in different portions of this new space, you can see that they cluster pretty well at different part of the space. And remember, PC2 was the component about tissue complexity. So if you look at the top of PC2, you see tracks that are more complex, like the arcuate fasciculus, which has a lot of crossings with other fibers, than tracks located at the bottom of the graph. For example, the uncinate fasciculus. This is a single fiber population bundle, or the GNU and the splenium. So it's kind of intuitive to interpret how these tracks fall in the space. But the nice thing about this space now is that for each cluster, one could, for example, compute the mean of the cluster and then compare each point to the mean by computing the Mahalanobis distance. And then this gives you an individual distance for each of these subjects to see how this person here, the splenium of this person compares to the mean splenium of all of the group in this new feature space that takes into account all of the different metrics. So that's a way of doing single subject analysis using multidimensional data. Now we're going to be looking at another way of doing it, which is with deep autoencoders. And we're going to be looking at two different autoencoders. So let's scale the data now using a min-max scaler, which is preferred for autoencoders. So again, we have our 216 inputs and 8 rows. So the idea behind an autoencoder is to simply take the data in, compress it into a latent representation, and then decompress it and compare the output with the input. And this should be the same. So basically by this compression, we're trying to learn only the important features in the data. And then if we specify that we want to encode it with 2 neurons, so in the middle layer, that means it's basically the same principle as with PCA, that we want to extract two components. And with every neural network, you can specify activation functions. In this case, if we specify linear activation functions, this is going to be replicating basically what we do with a principal component analysis. So let's first train our model and then fit the data and see that as we train, the error goes down. So that's what we want. We want to see this going down. And let's train quickly a second autoencoder, but this time using nonlinear activation functions. And then we're going to be comparing the output of both with PCA. So let's plot the previously generated graph and then the autoencoder 1 and 2 features for both linear and nonlinear. And then as you can see, each of them captures different information and data. So it's up to you then to see which technique you want to be using. But here are some pros and cons. So for PCA, of course, it's pretty fast. There's no parameter tuning. And of course, the problem is that it is linear. You can only model linear relationships between the features. Whereas with autoencoders, you can try to see if there is non-linearities between each of these features. But it requires more tuning. Finally, another way to do dimensionality reduction, which is pretty popular these days, is looking at UMAP. Fortunately, this is pretty easy again to implement. The thing with UMAP, however, is that it is mostly for visualization. So it is good to see different clusters, as we can see here. It's pretty nice, nicely separated. But the distances in this space or the organization doesn't mean much. So we have to be careful here with trying to do more quantitative analysis of these different points. It is a bit slower than PC also, but it scales pretty well. The more data you have, you're going to see it works pretty good. And again here, at least from the interpretation side, you can see that each of these clusters represent different tracks and they are nicely separated. Keep in mind here, we didn't input the tract label in the training. This is purely based on microstructural metrics for each of these tracks. Okay, so for the last portion of the talk, we're quickly going to go over anomaly detection. So before understanding anomaly detection, there's kind of a bigger overarching framework, which is called normative modeling, which is really trendy these days in the field, where basically you try to compare one subject against a group of healthy controls and try to see, is this subject part of the same distribution of the group, or how far is this subject deviating from the norm, for example. And this can actually be done with track profiles quite naively, because imagine you sample a group of healthy controls. Let's say you have 100 subjects, and you look at the mean apparent fiber density along the corpus callosum, and then you sample at 20 points, and you plot each of these subjects, so each of these purple lines here, as a normative distribution. And then a new subject comes in with a certain brain condition, and you do the same analysis. So you plot the points along the tract, and then you project it on this new space. And then you look, is this subject falling inside some sort of interval? So for example, 1 or 2 standard deviation from the mean. Or is this subject actually deviating from this? And this has been proposed and done a couple of times in the past. But it's actually quite easy way to compare each point along the tract to see which points are kind of inside and outside this confidence interval. So that's called the z-score approach. Now, as you see here, all these points forming this bundle. However, anomaly detection can also be done in a more multivariate approach. So a bit like we've seen before, if you were to project all of these points into a subspace, you could see how far is one subject from the rest using this Mahalanobis distance, which kind of takes into account more than a simple or single track profile and one measure for one tract. But we're going to be quickly going over another technique, which is based on deep learning, in the next slide. So a way of thinking about normative modeling could also be to train a network using only healthy controls data to learn what would be a healthy brain, basically. So if you give as input a set of track profiles which only come from healthy controls, and in this case we use the deep autoencoder. So we compress the data and decompress the data, and the goal is to obtain the same input and the output. And you do this and you learn basically what latent representation best represent this healthy control database. And then you give in a new subject, so one of your patients, for example, into this network, and you ask, what's the reconstruction error? So what's the output minus the input? And it might actually fall outside of the distribution if this subject has anomalies that the network has never seen before. Or it may actually fall within the distribution if the subject is simply as normal as the rest. So if you want to know more about all of these frameworks, there's quite a lot of them and a nice community around them. So there's the whole AFQ suite that has pretty good tools. I would also point you towards DiPi, if you want to work with Python code and diffusion in general. And there's also TractSeg, which is quite useful to derive these tracks. So with this, I would like to thank you for your attention.